Cellular transport is a method that cells undergo in order to get or maintain nutrients. And so what you're going to do is you're going to go ahead and listen to this flipped instruction and take some notes over cellular transport. So basically what you need to know is that every cell must have that homeostasis. It has to have that liquid environment that it needs to survive. That plasma membrane that we've already kind of discussed and done the lab with is selectively permeable, so that means that only certain molecules are allowed in and out. Diffusion is when particles tend to move from an area where they are more concentrated to areas that are less concentrated. In cells, substances can diffuse across membranes without requiring the use of energy. This is called passive transport. So looking here, we've got particles that are very concentrated. That means there's a greater number of them in a smaller area. And they want to be able to spread out and move into areas that are less concentrated. So you can kind of think of it as more room and more space. Osmosis is the diffusion of water across a selectively permeable surface. And membrane is what allows those cell membranes to regulate the cell's internal kind of atmosphere or areas. And the environment is where that homeostasis comes from. So basically, think about it this way. Osmosis affects cells depending upon the type of solution that they are placed in. There are three types. You have isotonic, hypertonic, or hypotonic. And so basically, if we kind of look at this, let's go ahead and look at facilitated diffusion, then I'll show you an image here in a second. But facilitated diffusion is basically where molecules cannot diffuse on their own. They have to move through those protein channels that are found in those plasma membranes. Active transport, on the other hand, is where transported proteins are carried through and it requires energy to move those across the membrane. It's almost kind of like against a grain, if that makes sense. And so it's kind of focused onto a different gradient. Endocytosis is where large particles are engulfed and then enclosed by the cell, and then they can move into the cell. Exocytosis is where large particles are released from the cell by secreting them or kind of expelling them out. And so if you were to look at these different types, this is diffusion, greater concentration to less concentration. Looking at this, if this were a selective or a semi-permeable membrane, once again, we've got greater, and we would be trying to move to lesser, greater here, and lesser. Okay, and so the main kind of focus here that I want you to look in on is that there are isotonic, hypertonic, and hypotonic. Isotonic, if we were to kind of look at the breakdown of the word, so isotonic means, and in Greek, iso means equal. Tonic's kind of another way of thinking of solution. Hypertonic means over solution, and hypotonic means under solution. So here is a nice little image. Sorry, that switched on us. This is a nice little image that kind of helps see those three different terms. So if I had an animal cell, so let's think to like our cheek cells for a second. An isotonic solution for a cheek cell or an animal cell is going to contain something that's along the lines of 0.9% salt. This is kind of, you've heard saline solution at a hospital. It's somewhere around the lines of 0.9%. If I have too much actual water in a solution, so it's hypotonic, then what ends up happening is that the cell takes in too much water because it's going from lesser to greater concentration. Example of a hypertonic solution is something like seawater that has a greater amount of salt in it. A hypotonic solution would be something that has little to no salt. And since animal cells do better under isotonic, a little bit of salt in there, this would be ideal for them. Too much salt would make the cell shrivel and shrink, and hypotonic would be very little salt at all, and the cell itself would take in too much water. Plant cells very similar to that. They also want to maintain this nice little homeostasis, and so just enough water, salt water mix that makes them happy. In a situation where they're in too much salt water or not enough water is actually helping that plant, they shrink and they shrivel and they don't look the healthiest. If I overwater my plant, I also put it into an hypotonic solution where basically it's just getting way too much water and very little salt, not in that perfect homeostasis. And so the vacuole, as you can kind of see here, is getting too big, and it's not good for the plant either. So we are going to use this information here in a second, and we are going to follow through with a lab.